Good morning students. Today we are going to perform the experiment of seeing the stages of mitosis in the onion root tip. Okay. So today's experiment, the aim is to prepare temporary acetocarmine stained mound of onion root tip. Okay. Now in this case we are going to study different stages of mitosis, right. For this we need the materials, means our requirements are onion bulb, conical flask filled with water, petri dish, scissors, forceps, needle, methyl alcohol, acetic acid, hydrochloric acid, acetocarmine, distilled water, this is microscope, slide, cover slip and blotting paper. All these are the requirements of today's experiment. Okay. The procedure I am going to explain you. See, before 5 days we had kept the onion bulb for growth of the roots. Now you can see clearly the roots are arising from the onion bulb. Okay. When we get the roots, after that what we have to do? We have to cut the 5 ml of the 5 millimeter of the root tip is being cut and kept in the solution of 1:3 acetic acid and methanol. Okay, 1 ml of methanol and 3 ml of the acetic acid. You can clearly see we had kept this solution for 1 hour. Okay, this solution is acting as a fixative. Okay, this fixative will you can clearly see the root tips present in the fixative solution. Okay, we had kept it for 1 hour. Now this fixative will fix all the stages of the mitosis. Okay. See, this is the fixative. Okay. Now this we had kept for 1 hour. In the summer season we have to cut the root tip at 9:30 to 11:30 am and in the it should be from. Okay. Now I am going to transfer this root tip, some of the root tips in 1N-HCl. You can see this is 1N-HCl. Now we have to hydrolyze it in 1N-HCl so that our roots can become very soft. I am keeping 4 or 5 roots in this. Now for hydrolyzation we have to heat the given, I will put all. Now we have to heat these root tips in 1N-HCl at 60° temperature. Okay students, for hydrolyzing the root tips we have to heat it at 60° temperature. Okay. This is the solution of 1N-HCl. Okay. Now we are going to heat it at 60° temperature for 15 minutes, 60° temperature for 15 minutes. See, by the process of hydrolyzation, the root tips becomes soft, okay, and we are able to make the slide very easily. can see the temperature is increasing. It's become 35 right now, but we have to heat up to 60 degree for 15 minutes. We can see this, our hydrolyzing solution is being heated up to 15 minutes at 60 degree temperature. Now we are going to take out the root tips and see the stages of mitosis in onion root tip. Okay students, now we have to wash these hydrolyzed root tips. I am keeping it in water, you can see I am washing them in water. After washing the root tips, we are going to keep it on the slide and cut the exit tip of the root. See, after washing, I am going to take on the slide, this is the root tip. Yes, you can see the root tip on the slide. Students, now you can see the root tip present on the slide. I have to cut the tip of this hydrolyzed root. This is the excess part which I am going to remove. This is the root tip. Now, with the help of blotting paper, we have to remove the water. Okay students, now you can see we are having the root tip on the slide. I am keeping 1 drop of the acetocarmine stain. This is your acetocarmine stain. Now this stain is going to be absorbed by the root tip. All the cells present inside the root tip. Acetocarmine stain is used to stain the chromosomes. See, we will keep it for 2-3 minutes and then we are going to use the cover slip to see these different stages of the mitosis. Now I am taking out the excess of the stain. Now our root tip is being stained. I am taking out excess of the stain and with the help of dropper I am going to put 1 drop of glycerine. You can see this is the glycerine. With the help of needle, we are going to make a 45-degree angle on the cover slip. Now, from the back of the needle or the brush, you can tap it so that the root can be split. I am removing excess of the glycerine. Now I am going to see it under the microscope. Now students, we can see the prepared slide under the microscope. We are going to see this under the microscope. We will be able to see different stages of mitosis. Yes students, at 10x we can see different nucleus having the chromosomes. At 45x we can see different stages of mitosis. We are able to see prophase, metaphase, telophase and anaphase in this slide. Now you people have to draw all these stages in your notebook and write this practical. Thank you.